Здравствуйте, уважаемые коллеги. Мы рады приветствовать всех участников.
Good morning, dear colleagues. Welcome to our webinar devoted to SARS-CoV-2 variants of concern, methods of detection. It's my pleasure to introduce Marko Marklevits to you. He is the first and the primary speaker for today. He's a laboratory expert from the WHO Regional Office for Europe, and he will be able today to present his presentation devoted to methods of detection and pre-screening for SARS-CoV-2 variants. Marko, welcome, and just a couple of your minutes. The presentation will be delivered in English. Therefore, dear colleagues, those of you who need translation, please select the Russian channel. I hope you do know how to do that. You need to look for a globe-shaped button below. Please push that button, select the Russian language, the Russian channel, and then you have to mute the original audio. Thank you very much for that. I hope we are clear with this. Marko, I guess the floor is yours now. Don't forget to unmute yourself.
Yeah, thanks a lot. Thank you very much, Laura. And also a big thanks for the invitation to present today. And I would start right up to share my screen. I hope this comes through. Can you see my slides?
Yes, we can see it.
Okay, great. Thanks a lot. So as Laura already introduced, this presentation will be around the detection of SARS-CoV-2 variants using SNP assays, and will also include the support that we are providing from WHO to Ukraine to implement these particular methods. Starting with a brief overview, I will start to give you some very basics on virus evolution. This is mainly for you to understand why SARS-CoV-2 is mutating and why variants actually occur. Then I will give you some details on the actual circulating SARS-CoV-2 variants. And as the third part, I will tell you something about how to detect and identify SARS-CoV-2 variants that will include how to actually detect them and including the principles and purposes of SNP assays, which will be then further explained by Laura. And I will put some special emphasis on the antibiotic SNP assays that allow the detection of SARS-CoV-2 variants. And I will give some next steps and outlook. So starting with the basics on virus evolution. So this is basically why and how viruses, in this case, the SARS-CoV-2 is mutating. To start with, the mutation rate related to the genome size. And let me see if I can. I'm not sure whether you can actually see my my pointer, but on the bottom right, you can see the higher eukaryotes, which somehow can also include us, but they are like higher organisms. So they have a very large genome size and a very low mutation rate. This is mainly because their genome is very complex, and mutations can have a very fatal impact. While when talking about the SARS-CoV-2, we are talking about an RNA virus, which is up here in yellow. So meaning the genome size is relatively small, and the mutation rate at the same time is relatively high. Sorry, the slides are not advancing. I just need to see. Oh. Okay. So related to mutations in the SARS coronavirus 2, what we have to accept is that the virus SARS coronavirus 2 is constantly mutating. This is a normal progress of the viral life cycle. But some genome regions within the SARS coronavirus 2 genome actually mutate faster. And the reason for this is that there is a higher selection pressure, meaning influences from the outside world to the virus to adapt to certain circumstances. And the gene that is mainly impacted here is the so-called spike protein, which facilitates the binding of the virus to the human host cell through the receptor binding domain. With regards to the mutation rate in SARS coronavirus 2, we can say that the mutation rate is half of the rate of influenza virus and 1/4 of HIV. And as a consequence, the SARS coronavirus 2 accumulates a two single letter mutation per month in its genome. This is just to illustrate with a very pragmatic, in a very pragmatic way, how viruses evolve. So what you can see here is a simplified human evolution, meaning a family tree of a human family, and the adaptation and evolvement of the genome actually is here facilitated through sexual reproduction, and with minor extent also by the accumulation of mutations. So viruses do not have such thing as a sexual reproduction, and thus they rely on other factors to adapt to circumstances. And one option is done also through mutations, and another option is via recombination. And what you can see here on the right-hand side is basically a family tree of SARS coronavirus 2 viruses, starting on the left-hand side and being more recent on the right-hand side. So the diversification of the SARS coronavirus 2 is pictured here in this, so to say, family tree of SARS coronavirus 2. As one fundamental aspect, I would like to give you an example with regards to the impact of mutations in the SARS coronavirus 2 genome. And on the very top, you see like a random extract out of the SARS coronavirus 2 genome with regards to the nucleotide sequence. And underneath are the translated proteins. So there are the amino acid get translated through the triplets of the nucleotides. So 3 nucleotides make up an amino acid. And according to the code illustration that you see on the right-hand side, different nucleotides make up different amino acids. And these illustrations you read from the center to the outside. So meaning a GGC sequence of nucleotides make up a glycine, as seen in the very first example here. So if the nucleotides then changes by adaptation of your mutation, it could happen that the position number 3 of the first triplet gets changed, meaning a GGA would still lead to the same amino acid, meaning that with the translated protein of SARS coronavirus 2, there are no changes. However, most important for us are the so-called non-synonymous mutations or substitutions. And this is the case if nucleotide gets exchanged that leads to the to a change in the amino acid sequence. And this is an example here. So from CCA, which would be then a proline, a CUA would lead to a leucine, so to a completely different amino acid with different properties. And translated into a protein, this change may facilitate a change in function or adaptation to certain to certain elements. For the sake of completeness, most of the mutations that actually occur within the SARS-CoV-2 genome are lethal. So that means that the virus cannot actually survive and will eventually die. And one good example is when an amino acid changes to a stop codon, which is shown down here. So UAU was changed to UAA, which results in a stop codon and thus in a truncated protein, which is then in most of the cases not functional anymore, and the virus cannot replicate further. So this is important to highlight. Most of the mutations in the SARS-CoV-2 genome are actually lethal for the virus, so they don't survive, but it's continuously occurring that the virus is mutating, and some of these mutations are eventually beneficial for the virus to adapt to the environment. Coming to the second part of my presentation, which will be around SARS-CoV-2 variants. And we call them variants when they actually have established a so-called advantageous mutation, meaning a mutation in the viral genome that leads to a benefit for the virus, for example, to be easier transmitted or to replicate to higher numbers. These are two screenshots that I took. So the first one actually I took for a presentation at the beginning of June. And these colored lines show the frequencies of lineages of SARS-CoV-2, meaning genotypes, different variants of SARS-CoV-2. And as you can see, from the very beginning, there were some dominating lineages, which were called A and B, and they somehow disappeared over time, while others then got an advantage and replaced other lineages. And that's the case actually for the B117, which is the alpha variant that has been first identified in the UK. So around April this year, this was dominating half of the viruses that were present worldwide. And as you can see here with these these labels on the right-hand side, there are also the beta, gamma, and delta variants in minor proportions. But what you can already see is that the delta variant has already quite some proportion of the overall lineages globally. And when you have a look at the same lineage distribution a bit later, which was actually the day before yesterday, so 14th of July, we can see that the B117, the alpha lineage, which is this one, so this time a bit lighter in blue, is somehow reducing, and it gets replaced by the delta variant. And this means that the delta variant may have some advantage over the alpha variant. So even though at the time point, beginning of June, where the alpha variant was still increasing over time, now there's a new variant with a more beneficial set of mutations that replaces then the alpha variant. So why is it actually important to have a look at SARS-CoV-2 variants? And that the reason for that is how fast, for example, a virus can transmit in the population. And that's important to mainly introduce public health measurements that can help to reduce the transmission rate. Because for the Delta variant, which is actually this curve here, very steep up, means that within a shorter period of time, the number of cumulative cases actually increases drastically. And this means that the virus is possible to infect more cases in a given amount of time, which then requires the need to implement more strict public health measurements to control transmission. And that's the reason why we need to monitor our SARS-CoV-2 variants very carefully, so that we know when our measurements that are currently in place are not sufficient to slow down the transmission chains or the infection rate in general, and which has then an impact on our public health system. WHO has defined some working definitions for SARS-CoV-2 variants, and they're not preliminary anymore, so those ones are the final working definitions. And WHO considers a SARS-CoV-2 genotype as a variant if it fulfills certain criteria, and those are listed here. So if a SARS-CoV-2 shows actually a phenotypic change, and the phenotypic change is a change in behavior that could be transmission rate, that could be morbidity or mortality, or that this is associated with an amino acid change that I have explained earlier, that is already known to establish or suspect some changes in behavior in the virus. And one of the arguments down here, so either that has already established a community transmission or multiple cluster, or if this particular variant has been detected in multiple countries. Or, and this is an optional second assessment, WHO has established a so-called virus evolution working group, which on a regular basis assess signals of SARS-CoV-2 variants globally, and that are likely to have a stronger on particular virus can also be declared a variant of concern. With regards to tracking SARS-CoV-2 variants, I would like to point you to a website.
Marco,
which
I cannot hear you sometimes.
Down, and you also.
Yeah, I cannot hear you sometimes. You're disappearing from time to time. Sorry. I can't hear you at all now.
Okay, let me just see whether I can.
It's like you are dropping out for something like 3 or 5 seconds, then you appear again, then you drop out again. Marco, are you there?
Apologies for that issue. Do you hear me now?
Yes, I can hear you now, but it's not perfect. Can you like continue talking? Maybe it is fixed now.
Коллеги, пока Марко решает технические вопросы, связанные с соединением.
And Laura says now, as we are now trying to fix this technical issue, I would like to remind you, dear colleagues, that you can ask your questions with the use of the chat box, and we will try to give the answer by the end of the presentation, in the end of the presentation. Please be informed, we have simultaneous translation available. For that purpose, you have to select appropriate language, either English or Russian, depending on your preferences.
Apologies for these technical issues. Can you hear me now clearly? And,
yes, I can hear you now. Yes, we can see it, and I can hear you. Thank you.
Marco, thank you.
Okay, perfect. Apologies, that was very unfortunate. Did you lose me earlier, or can I continue here?
A bit earlier, if you can go back to the previous slide, please.
I would just continue from this slide. So this is about how WHO WHO actually tracks SARS-CoV-2 variants. And there are two types of variants that are so-called variants of concern and variants of interest. So variants of concern is the higher classification, meaning that these particular variants have a higher impact on the transmission, while variants of interest are getting monitored and eventually being declared a variant of concern at a certain time point. And I would like to draw you to this website that is listed down here. You can also use your cell phone to scan this QR code, which directly would lead you to the respective website at WHO. And the table which I'm showing here is just an extract showing the variants of concern that WHO has declared, meaning the alpha, beta, gamma, and delta variant. And for you, or just to highlight now, is with regards to the fifth column with regards to additional amino acid changes. So a variant can also include additional mutations over time, which then would still fall into the same umbrella of the WHO label. For example, the 484K mutation or the 452R mutation in the alpha variant. So this website gets updated regularly depending on WHO assessing certain variants as a variant of concern. So coming now to the actual variants and their distribution globally. So what you can see here are the distributions of the different variants of concern, meaning alpha, beta, gamma, and delta globally. And what you can clearly see is that alpha is basically present in every country of the world. So is also beta, with the exception of Northern Africa and parts of South America, while the gamma variant is actually a bit less widely distributed, completely excluding Africa, and the delta variant is actually increasing globally, and it has almost the same coverage as the beta variant reached over time. So the last part and the most important part probably of this presentation is how to detect and how to identify SARS-CoV-2 variants. And with regards to the technical aspects, I will keep my presentation relatively short, as we will hear now or soon from Laura a bit more details on that. So how can variants actually be detected? And the most obvious method is to perform whole genome sequencing. But as we all know, this is a quite expensive and labor-intensive technology that is not available in all the countries in our region. And with regards to the lead time, meaning the time from taking the sample and having a result on hand, there's a certain delay of at least 1 week. So there are other methods that are faster in delivering results. And one method is to perform Sanger or just partial high throughput sequencing, meaning an amplicon-based sequencing of the gene of interest. And when using Sanger sequencing, the results are earlier available than when using high throughput sequencing. There's another method called RFLP, which basically uses enzymes that cut a certain signal sequence which is related to the mutations that you would like to identify. And depending on this restriction pattern, you are able to identify variants. The fourth method is actually quite well known, in particular with regards to the identification of the very first variant globally, which was the alpha variant identified in the UK. And it was identified during routine surveillance and the observation that 1 out of 3 real-time PCR targets was systematically failing. And the reason for that was a deletion in the N-terminal part of the spike protein, And due to that, it represents a good proxy to identify or to give a strong hint to the presence of an alpha variant. Another option is, of course, the use of the real-time RT-PCR, meaning, for example, multiplex PCRs to identify several mutations and potentially representing different variants. But this technology requires a bit more of technical background also with regards to the requirements to real-time instruments. And due to that, the last point on this slide, the single nucleotide polymorphism or SNP assays are quite a very handy tool to identify SARS-CoV-2 variants. And the reason for that, I will tell you in the next slides. I will keep this very brief, and Laura is likely elaborating a bit more on that. But the identification of SNPs of single nucleotide polymorphism, meaning single nucleotide exchanges in a sequence that lead to an amino acid exchange that then may have a beneficial impact to the virus, is done by using probes that are actually having the target of this particular SNP. And it works through the different energy, and this is now here represented as temperature that you need to invest to separate the nucleotides that are binding each other. So G to C and A to T, they have different strength in binding affinity to each other, facilitated by these hydrogen bonds here between the G and C. There are 3, and between A and T are 2, meaning that in this probe sequence, you need a different temperature, you need to invest a different amount of energy to actually dissociate these probes. And this is the chemistry behind what we use in SNP assays. So in the SNP assay, there is a constant increase of temperature and measuring of the fluorescence. As we know from regular SARS-CoV-2 to real-time diagnostics, it's just that the measurement is done after every increasing step of the melting temperature. And this will then eventually lead to a so-called melting curve. And this indicates the time point where 50% of the probes have dissociated from their targets, and that allows the discrimination of the different variants. So with regards to our support from WHO side, and also now with regards to the support that the company that produces this particular assays, Tipmobile, is providing to Ukraine. So we have firstly have a rollout of this first assay targeting the N501Y mutation within the SARS-CoV-2 genome. And this particular mutation was associated with 3 of the now 4 variants of concern, meaning alpha, beta, and gamma. But since we know now that this mutation has become a very dominant one, we needed to come up with something else to detect or discriminate also the different variants of concern. And the additional support that now is done by Tipmobile through the donation of kits to Ukraine, through these 4 additional assays that we are now providing. And these assays target different key mutations that are characteristic for the different variants of concern. And the amino acid that is targeted is shown in the second column here. And as you can see in the rest of the table, different mutations are present in different variants of concern, so that the identification of these mutations allow a proxy to predict the presence of a variant of concern. Just to give you an example, because it's it's a new new approach that, for example, at the position 417, there are two different amino acids present, meaning T and N, depending on the two different variants. So within the beta variant, at this position, there's actually an N present. And within the gamma variant, there's actually a T present that replaced the K, so the lysine, at this particular position. And with this detection, you can actually discriminate between the beta and the gamma variant. Within the alpha and the delta variant, this mutation is not present. It still has the, let's call it, wild type sequence, meaning the K at this particular position. Just want to highlight the last assay, assay number 5 here, which is an exclusive marker, so the T478K. It's a particular marker for the presence of the Delta variant, which is probably right now the most relevant one. But this set of SNP assays allows to type the presence of different variants of concern within the sample set. This is just to give you an example how it then will look like in the laboratory if you test the sample with one of the assays. And this is now the example of the K417 assay. So you have a control, as you have in all laboratory assays, that shows you the amplification of the target sequence. And in the melting curve analysis, this is a picture that the real-time machine would produce, and that gets translated in the melting peak that I had mentioned earlier. So with regards to the lysine, so the K amino acid at the position 417, that's the peak here in the middle, and this represents the wild type. So if there is a shift in temperature, meaning shift in the peaks to a lower temperature, this then represents an N at this position, so predicting the beta variant, and shifting to a higher temperature, meaning a T at this position, which then has is a proxy for the presence of the gamma variant. So at the same time, one particular SNP assay can discriminate between the wild type, the beta, as well as the gamma variant. Now I want to show you, for those who are not familiar with the N501Y SNP assay that we have provided to Ukraine earlier, and the Delta assay will be delivered in the same format as you see here. So that's analog to the Delta variant assay. And so the kit is very much simplified so that it can be used in basically every laboratory that is performing SARS coronavirus 2 diagnostics. So the amount of or the number of tubes and reagents in this kit has been limited as much as possible. So it basically contains 3 different tubes. The first tube is actually containing a buffer, which can be used then to add to the reagent tube. And let me just explain you this particular reagent tube a bit more in detail. So you can see it here on the right-hand side. And the tube contains a pallet at the bottom, which is here in white. And particular part are the primers and probes that are needed for the assay. Can you still hear me? It shows me that my connection is unstable.
It's a bit unstable, but I still can hear you.
Can I just ask the translator to confirm that you still hear me?
Yes, I can still hear you. Marco, I can still hear you. It's okay so far. Please continue.
We hear you and we see you, Marco. You can continue. Thank you.
Okay, thank you. Please just interrupt me if the connection keeps breaking, then I will try to solve this problem. So the instruction for use is as simple as we could do it, in a way that you just transfer from the very first tube with a buffer 800 microliters into this special tubes, which solubilizes the polymerase as well as the primers and probes. And then you're actually already done with your preparation, and you can dispense this mixture into a 96 well plate. So we use 8 microliters of this mixture, and we would add then 2 microliter of RNA of the pre-tested sample. So meaning the RNA sample that has already been made available after extraction during the routine diagnostics. So by adding 4 microliters, and then this the mixture is already prepared to be then inserted into your real-time instrument and to perform the assay. So the benefit is also that you can freeze this mixture at -20 if
I cannot hear you.
the complete 90 tube. And the reason why we have decided for lyophilized polymerase and primers and probes is the fact that this kit can be transported as well as stored at room temperature. So we don't need any temperature controlled shipments to transport this kit to laboratories or into country. This is just an example with regards to the thermocycler configuration, and this should just show you that this is a typical PCR protocol with the different steps, like a denaturation and annealing extension temperature, and this is done several times, as you can see here. So this is an example of the RotaGeneQ software when programming it. And this is the step, the very first step that every SNP assay has to do, which is basically the amplification of the target. And the success of the amplification can be checked by using the amplification curve. So there's also a probe included that indicates the amplification of the target. And that's your quality control, so that you know that you have sufficient starting material to perform then the melting curve analysis. This slide shows you then the program of the melting curve, which is shown here on the right-hand side. So as I said previously, the SNP assays relies on the binding affinity of the probe to the target sequence in the SARS-CoV-2 genome. And depending on the presence of a potential mutation, this temperature can differ. So the real-time instrument is sensing this difference by slowly increasing the temperature over time during the melting curve analysis and performing a measurement after every increment step. And as a result, you will receive these melting curves, which have then a characteristic dissociation temperature. And just to highlight this again, so this peak actually shows where 50% of the probes have dissociated, so detached from the template. So at 59 degrees, the wild type, so the 501N is indicated, while an increase in the melting temperature to 64 degrees, the 501Y mutation is suggested. With regards to the stability of these assays, it has been validated with regards to freezing and thawing. As I said, non-used material from the mixture prepared can actually be frozen and thawed after up to 3 times without any quality loss. And so an amplification is possible until the CT value of 38, which is the equivalent to approximately 2 copies, so very stable and sensitive assay here. And the melting curve will be interpretable up to a CT value of 34, which is an approximate equivalent to 40 genome copies in the sample. With regards to the compatibility, this was one of the main issues that we would like to have addressed properly in these assays, and that's the compatibility to a wide range of available real-time instruments. So the idea was that these particular assays can be run in every laboratory that is performing SARS-CoV-2 diagnostics. So those instruments that are listed here, so the rotagene, QIAGEN rotagene, the Bio-Rad CFX96, and the two Roche instrument, as well as the ABI QuantStudio, represent the vast majority of instruments and that is present in the different laboratories. However, apart from these 5 instruments, it's expected that these assays can be run on a wide range of additional instruments. And the background of that is that with these assays, we just rely on one single channel that we need to be readable in this instrument, and that's the FUM channel. With regards to storage and transportation, I have already mentioned this. Due to the lyophilized format, this assay can actually be transported and also stored between 4 and 25 degrees. And the assay is even stable during transportation up to 45 degrees, which does not require any temperature controlled shipments. We have to keep in mind that we can store the assay at 2 to 8 degrees for a short period of time if it will be used, for example, a bit later during the day. But if you don't use up all this mixture, we kindly ask you to freeze the remaining mixture at -20 degrees. One thing we need to highlight here is that the assay in a lyophilized format, so in the status that you receive it in the laboratory, please do not freeze the lyophilized assay, as it will damage it. And as I have mentioned before, so freezing and thawing of the solubilized reagent is possible up to 3 times without any quality loss. Last but not least, I would like to give some outlook and also tell you about some next steps after a SNP assay positive sample. So we need to keep in mind that a positive sample is very likely to be associated with a variant of concern. However, it will not be a proof, since we are just relying on a single amino acid exchange. By using these 4 assays that we are now providing to countries, we can make use of a second assay to give a more, to increase the likelihood and the confidence of this detection. However, the national testing strategy should actually define what the next steps are after a positive SNP assay result. However, these indications given by the SNP assay should be considered as a valid identification of the presence of a variant of concern. What we would recommend as optional steps is to perform sequencing of at least a proportion of the positive samples detected with SNP assays, and that can be done in the ways that I have mentioned earlier with regards to partial sequencing, performing Sanger sequencing that is more widely available than high throughput sequencing. And this can be done by targeting the most important gene, so the gene that we are also targeting with the SNP assay, meaning the spike gene, which is also including the receptor binding domain. But of course, the gold standard to follow up and to get a good overview on the diversity of sequences in your sample is to perform whole genome sequencing. And Ukraine is currently building up in-country capacity, but we have also supported Ukraine to liaise with other organizations, in this case, Public Health England, to also receive some support in sequencing. However, we still offer from WHO's side the support through our reference laboratory network. So laboratories in Ukraine can request support to send samples to one of our reference laboratories to perform sequencing of the sample. There's also the opportunity to request support through the GISRS network. This is mainly can mainly be done when also surveillance sentinel samples for SARS-CoV-2, so COVID-19 and flu, if they will be included in these shipments. Of course, there's always the option to send samples to commercial sequencing companies, but this has not really been an option for many countries. So what we would highlight with the last point here with regards to an outlook, we are looking into the second rollout of now SNP assays to countries, and we would like to facilitate a continuous support on that, so that if new variants occur, that we can then provide adapted SNP assays to the country. And that is shown in my last slide here. So the different mutation patterns of the variants of concern allow us these typing of particular variants. And if there's a new variant of concern being declared, we will be relatively fast with a special acknowledgement to the support through TIP-Mobile, which use very short time to develop new assays targeting then a new potential key mutation that is characteristic for a new variant of concern that may emerge in the near future. And with this, I would like to thank you for your attention, and I'm readily available to answer questions. Back to you, Laura.
Марко, спасибо большое за вашу презентацию.
Marco, thank you very much for this presentation.
Я также хотела поблагодарить компанию TIP-Mobile за предоставление.
I would also like to thank the TIP-Mobile company for supporting Ukraine with the SNP assays. We have been provided with a batch of these test kits of these assays in spring and just recently, and this is what we're grateful for, definitely, as well as to the WHO, which also participated in organizing this support. We don't seem to have many questions at the moment, so I suggest I go to my presentation maybe, and then we will be able to talk about all of these questions at the same time. So let me just start sharing my screen.
Одну секунду. Я прошу прощения, я сейчас пытаюсь настроить, расшарить свою презентацию.
Give me just a second, I'm trying to start sharing my screen and I still cannot do that. Sorry.
Вот теперь. Скажите, пожалуйста, коллеги, видна ли моя презентация?
Colleagues, do you see my presentation?
Yes, we do, just not in presentation mode yet.
Хорошо. Секунду. Не в режиме презентации, я поняла. Так. Прошу прощения. Так. Коллеги, я предлагаю, вероятно, у меня какая-то проблема техническая с отображением. О. И теперь в режиме презентации.
And now, can you hear it now, colleague?
Yes, we
Да.
can see it full screen.
Спасибо. Спасибо большое. Итак, уважаемые коллеги.
Okay, thank you, colleagues. So with your permission, let me tell you the following. Most of you, I guess, got used to using the real-time PCR for the sake of visualizing the amplification products with the use of amplicons. But you know, some special probes that can be used for that purpose. On the one hand, that improves the specificity of the assay because they are specially selected for the unique genome fragments of the target organism. That system allows you for multiplex analysis. You can focus on different zones. You can dye probes with different types of these dyes. On the other hand, that makes the situation, that makes this process more complicated, especially when the organism genomes are not very different, or in case of an SNP mutation, when the amplified fragment can be different by one letter only. What the TIT Molbiol company suggests and the type of assay that we talked about today in the Marcos presentation is a bit different visualization method. The connection is very poor, I'm sorry. But this is something that we can discuss, and this is the one that can be used. I am sorry, the quality of sound is not good. Non-specific complementary probes and dyes can be used for that purpose. Sorry, no sound again.
Коллеги, я прошу прощения, вероятно, связь прервалась.
I am sorry, dear colleagues.
В наборах реагентов, основанных на принципе анализа кривых плавлений, используются
the assays using the melting curves principle, they have a different approach. This is the intercalating dyes. These are the special substances able to get into any molecule, increasing the fluorescence, two-string molecule. That is quite convenient because this intercalating dye is pretty cheap. There is not many difficulties in selecting the one that you want. On the other hand, they are just a bit limited with regard to their multiplexing. Secondly, they can get into any two-string component, including the specific or non-specific one. For that purpose, for you to be able to differentiate the necessary target amplicon from the non-specific products of amplification, you might need an additional step, the so-called melting curve analysis. So what happens during this process? So let me tell you that to have this additional stage for the melting curve analysis, you do not need any additional special equipment, no special needs to organize the working process in the laboratory. It all happens in your RT-PCR machine. That might be either a separate stage of the process, or it can be included into the currently available process. Basically, it looks like an additional step into the amplification program. After the well-known cycling process for you, during which the temperature starts for the sake of elongation, quenching, etc., then you add a so-called melt curve analysis into the program. At this time, we see that the tube is getting heated to a high temperature bit by bit until, let's say, 95 degrees centigrade or whatever you indicate. And every half a degree or even less during the tube heating, there is a result taken from the point of view of the fluorescence intensity. So what is happening in the tube? We remember that the intercalating dye gets into the two-string molecule of DNA, any of them. In the preceding cycling process, we have developed a certain amplification product. The intercalating dyes have been included into that. So what happens there? You know that the intensity of fluorescence gets higher, which means in the tube we see the high intensity fluorescence. So what happens next? Then we start heating the tube bit by bit, and we collect the fluorescence results. And what is happening to the DNA when you start heating that? Definitely it starts dissociating. Sorry, the quality of sound is much to be desired, but the association and the links get broken. The intercollecting dyes also leave the two-string molecule, and fluorescence intensity goes down. Please take a look to the left part. That looks like a typical fluorescence measuring in the testing tube. You can see that in the very beginning of the melting curve analysis, the fluorescence intensity was pretty high. Then, at some point, there is a very steep drop in intensity. This means that there was a dissociation of the hydrogen links. There was a denaturation in this process, and you see that fluorescence intensity went down. Could you tell me? Sorry, I do not hear the question, but the continuation is what would be the temperature for melting? Sorry, guys, I asked you a question, but you do not have the right to unmute yourself, and I will give the answer to my question. It will very much depend on the composition of the amplicon. You might remember from the Marcos presentation, there was a slide with complementary pairs, A and T, G and C. Adenine, thymine, there is only 2 hydrogen links, and G and C pair is more tightly related, so to say. And to break these links, you need to attach more energy or higher temperature. This means the more G and C letters you have in your amplicon, the more temperature you need for denaturation to make sure that these 2 parts of the DNA get broken. That means you will see a higher temperature when the fluorescence intensity goes down. We know the amplicon composition, and that helps us a lot. Let's now imagine the following. We have 2 variants of SARS-CoV-2. One of the variants has got SNP change. The T in the genome was substituted by the G. That means that the melting temperature will also change. Because T was substituted by G, the number of hydrogen links has actually increased in the amplicon. This means the melting temperature will be different compared to the amplicon of the wild virus. So this is the basic principle for melting curve analysis. You basically have to look for the temperature at which the dissociation of the intercalating dye took place, and the fluorescence intensity drops down significantly at that point. If you see a specific necessary amplification product, and you see the drop down in the intensity of fluorescence, this is what you expected. If it is not specified, sorry, the quality of sound is much to be desired, but the continuation is as follows. And this is also true for variance detection. So if we observe fluorescence intensity dropping down at 58 degrees Celsius, that means in the amplicon you see a T which complies with the wild type, for example. If we see intensity drop at 64 degrees Celsius, just for example, that means the number of G and C, sorry, the quality is much to be desired compared to the wild type. Let's now get back to our chart and look to the right part of it. It is not very convenient, really, to simply observe visually the intensity drop in fluorescence. It is not very convenient and not easy to follow, definitely. Therefore, you should use some special tools for that. You should use a certain rate for that. This rate should be the rate of fluorescence drop. So, in other words, the quicker the rate drops down, the higher would be the peak of this rate on the chart, and the top of the peak would comply with the temperature. Quality of sound is much to be desired, I'm sorry. Pay attention, it does not correlate with the concentration of the target in the tube. If you see 2 peaks, one is higher, the other is lower, it does not mean that the concentration of the targets in the tube was lower in the higher and the lower peaks. Let me just remind you that these peaks are actually the rates. This is a rate that tells you about the change in the intensity. So basically, to what extent the intensity rate dropped down or changed in that given tube. Definitely, it is not that easy. You can hardly find perfect peaks. You can only find them in really perfect test kits. Sometimes you can come across situations which are not easy for interpretation. I suggest we go on talking about this issue only with regard to the peaks, these peak rates. We do not speak about the curve in general. So if the sample contaminated with the genomic DNA, you might see additional peaks on the picogram. So how do we realize which one complies and correlates to our product? Is it contaminated or not? To do so, you need to have some control samples. For example, negative extraction control. It must be negative, of course, because it should not hold any peaks in all channels of detection, but for the channel which you use for the sake of internal control. Moreover, you can use a so-called no reverse transcription control, which is the one that does not go through the reverse transcription stage. So in this case, it will have a positive peak. If it is a clean example, then the no reverse transcription control will not have any additional peaks. Speaking about the positive control for amplification, can help you to get rid of such BS or failures like the primer dimers. What is a primer dimer? This is the one that can hold complementary, the primers might hold complementary regions. So basically they can stick to each other, and the polymerase might build up additional complementary fragments, making up non-specific products for amplification. And they can give you additional peaks on these graph, and sometimes might collate with the target amplicon and BS that. What can be done to visualize these primer dimers and cut them off? You need to use the NTC. But here, this is the one where you see the primer dimers more often, because in the negative primer control, you do not have any other target, and these excessive primers can build up. And in the normal biological samples, you usually don't see that. But still, you must use the control for sure. Additional peaks on this graph might not always be associated with contamination and formation of primer dimers. They might be associated with the qualities of the primers or amplicons. We expect that during the melting curve analysis, the amplicon melts down in a very linear way. So we imagine like there was a kind of an amplicon, and at a certain temperature it gets denaturated completely. But in the real life situation, no, it doesn't happen like that. In the real life situation, there are some regions which are more GC rich or less rich in GC. And dissociation of such an amplicon happens in several stages. Under low temperatures, we usually see dissociation of AT rich regions, and then at the high levels, we observe the GC rich areas dissociation. Therefore, you might see additional sub-peaks on the graphs. Well, that was very briefly from my side with regard to the mountain curve analysis. If you have any additional questions, I would like to I will try definitely to give answers to your questions. And if you want, we can now look into a real life protocol for programming. I can show you how to uh, initiate the mountain curve analysis program if you wish to do so. This is it from my side, dear colleagues. Thank you very much for your attention. We are ready to give answers to your questions, if any. Okay, question number one. It seems like Marco already gave the answer to that question. Let me actually voice it out. Is it possible to send the N501Y SNP assay to Moscow? NIC, I do not know what is that laboratory actually, and what should we do for that? And the answer from Marco is yes. We can provide you with a limited number of these assays, and he asked to kindly share the email address. I think that question is quite a relevant one to other colleagues. If you wish, if you feel like you would like to get these essays, if you are staying in Ukraine, you should address that issue to the WHO country office in Ukraine or directly to the regional WHO office for Europe. Somebody says, show me the protocol. What exactly do you mean? Because I can show you the instruction and I can show you the protocol.
Вот так выглядит русифицированная инструкция, которую мы
This is the way the instruction for use looks like. This is the one that has been translated into Russian. It is also available in English and Ukrainian as well for VIR-SNP N501 501Y. So what they suggest, you basically have two options. In fact, you can perform the normal PCR, and then in the same tube you can do the molten curve analysis with the use of additional software, or you can combine the protocol, you can combine the program, and the first part of it would be just a common PCR, including the standardized cycling, reverse transcription in the beginning, 55 minutes, I guess, then the hot start. You have to activate the ferments for reverse transcription and activate the polymerase. Then cycling, 95 for 5 seconds, then 60 for 15. This is primer quenching. And then you see elongation. The number of cycles might be 40 to 45. You can it can develop depending on the number of copies in the sample, or if you are limited in time. For example, if you have a shorter amplification program, for example, you can use 40 cycles for amplification. If you want to get more sensitive result, you can get additional 5 cycles. And then cooling down for 30 seconds. This is the first part of a very standard PCR. Then you add the melting curve analysis. For the well-known Biorad CFX96 for you, there is a special screen, protocol screen here. So basically what you do in the first stage, you indicate the following. You see 95 degrees for 5 seconds, 40 degrees for 30 seconds, and then you select the melt curve analysis, 45 degrees to 73.7, increment 0.7 for 5 seconds, and then there is a plate read every on each increment. So basically you indicate the initial temperature and the final temperature, and then you indicate the increment. And it basically prolongs the total program for about 20 minutes. The only thing you need to do is to add these additional steps to your program, and that's it. So speaking about the way the real live protocol looks like, I cannot show you the protocol for titmol-biol run. I don't have it now with me. I can show you something very similar using the same principle, just as a... I will show you the one now. Here we had several detection channels. In titmol-biol, this is only the FAM channel used. I will show you the Quasar 760. To the left, you see the fluorescence measuring in the tube. You see how it goes down under the temperature of 60 to 63 degrees. And here you see peaks on the melt peak diagram. And you also see the results based on the temperature at which these intensity dropped down, and you can see it in the table here. And you see, for positive samples, it was 63 degrees. Those that didn't have peak, they are listed here below. It looks pretty simple. Another question, do you need to have additional paid software for HRM, or just common melting is enough? HRM is available into the common soft of RotorGene if you use the RotorGene. Curve analysis, melt curve analysis can be added to some other pieces of equipment. Some companies suggest the interpretation software for that purpose. It is pretty convenient because you immediately can see using that software if it is positive and negative. There is no such interpretation software for titmol-biol program as far as I know, but the results that you obtain with this titmol-biol product are pretty clear. You have peak, then it's positive, no peak, it's negative, and definitely you can see from the temperature, from the melting temperature, what is that type of variant. And this is it. This is very simple. Colleagues, do you have any other questions? Let me remind you that we have a recording of the webinar. Therefore, all the materials, all the records will be circulated to you if you want. You can also apply to me, to Marco, to the organizers. If you have any questions, you can ask them additionally later on. And we will definitely try to give answers to your questions. I actually don't see any more questions on the chat. So colleagues, I give you another minute. And then if we don't have any questions, then we are done. I would like now to express my gratitude to Marco, to the WHO Regional Office for Europe, colleagues for organizing this webinar. Thank you very much for this beautiful presentation, and thank you very much the Team All company for supporting Ukraine with these type of essays. Thank you very much. And dear attendees, dear participants, thank you very much for attending. And I see from the chat that everyone is very much grateful to you, Marco, for your beautiful presentation. Marco, do you want to say a couple of wrapping up words? Then you are welcome if you want to do so, because I'm ready to close the workshop.
Thank you very much, Laura. No, I'm very happy that the presentation has been well received by the audience, and I'm readily available to also reply on questions that you may have after this this webinar. So please feel free to reach out either to me or to Laura or to the organizers to address your questions, and I'm very happy to follow up with that. Also, with regards to requests to WHO to provide you with the SNP assays that I have mentioned, we will put out a call to express interest in the probably next week. But please feel free to already write to me or to the colleagues in Ukraine, so that we can plan accordingly to ship these assays to your laboratories. So with this, it just leaves me to again acknowledge the opportunity to present here. So thanks to the organizers, also thanks to the interpreter in Ukraine for this help, and also for all the participation here during this meeting. So thanks a lot for that.
Спасибо большое, коллеги, за участие. Тогда мы закрываем.
Colleagues, thank you very much for your participation. I think this is the end of our workshop.